Hello, everyone. This is Indresh, and I'll be moderating the analysis and visualization tool. There are three speakers, starting with Eneida Hatcher from NCBI.
Hi, I'm back. Eneida Hatcher from National Center for Biotechnology Information at NLM, and I will be talking about NCBI virus and some of the resources that we have at NCBI that are relevant to this discussion today. So to start, as you are doing analyses and trying to make decisions about how to help public health, how do you find virus data that has certain characteristics that fit your usage needs? What if you need data from a certain geographic location or that was collected recently? What name do you look for? As we've had lots of discussions today about the variability in names that you can use. What if you're looking for part of a lineage or for a subtype? We have, well, maybe. There we go. Nope. I'll talk through and see if it comes back. We've developed a resource called NCBI virus. So this gathers information from various resources at NCBI, including GenBank, which is part of the INSDC collaboration. So they have data from DDBJ, ENA, GenBank. They all get merged together overnight and they're available through each other. So we pull data from GenBank. There we go, which includes the sequence data itself along with metadata describing the sequences. We also pull data from records called BioProject and BioSample. So those aggregate information about research projects and research samples. So they have additional metadata that we can incorporate. And we're starting to explore pulling in data from SRA. So that is the NCBI resource for unassembled raw sequence data. But that also has a lot of information that's unique, right? So minor variants that might be present there. We pull all of that together, link all of the information so that it's coming in one place. You can find that through NCBI virus. And then on top of that, we add in information from RefSeq and from Taxonomy. So taxonomy is one of the primary ways that we find that people want to look for the information. So we bring in NCBI taxonomy to organize all of the sequence information. We also curate the metadata that's associated with the sequences. So for example, somebody makes a submission to GenBank or one of the associated resources, and they can say that the sample came from serum, from plasma samples from HIV or HCV co-infected patients, from plasma, from PBMCs. If you had to search for every one of those terms to find what you were looking for, it would take you all day. So many headaches. So we normalize all of those terms to something that's a little bit more manageable. So in this case, you could just look for things that came from blood, and you would capture all of the records that have this type of information associated with them. So we have some of the other fields that we do this kind of normalization for, shown on the screen. The host, the sample location within the host, geographic regions, lab or vaccine strains. We look for certain terms and mark up the records with those terms. And then all of this is available through the NCBI virus web interface. So it makes it really easy to use filters to sort through a results table where you can see all of the metadata that's associated with the sequences and then download the sequences also from that same resource. And it has very flexible downloads. So if you want the actual sequences, if you want the metadata, if you just need an accession list, it's all very easily, hopefully right at your fingertips. We're also responsible for creating RefSeq records for viruses. So RefSeqs are representatives of a species or also of an unclassified group, and I'll get to those in just a moment. They are based on GenBank records. So basically we pick a GenBank record, or a GenBank record is picked, and that can get copied and created as a RefSeq, which we can add updates to. So we can improve the annotation. We can add references like publications. We can add source metadata that might not have been provided with the submission, but might be available through a publication. So we basically try to make them as comprehensive as possible. Let's see if, there we go. Okay. Usually we try to have one RefSeq per species, but there are many cases where it makes more sense to have more than one per species. So for example, SARS coronaviruses, right? There was one for the older outbreak. It wouldn't make sense to not have a new RefSeq for SARS-CoV-2. So that one has multiple RefSeqs. Also, we'd create the RefSeq records based off of ICTV exemplars. So those were mentioned earlier. Occasionally, sequences will come in that don't fit into ICTV recognized taxonomy just yet, right? The taxonomy just hasn't been reviewed. It hasn't been created yet, but we think that it's still an important virus group. So we want to have a reference available for it. So we'll create a RefSeq based off of something that's early, something that's complete, something that looks representative and has good metadata associated with it. Also for well-characterized viruses. So these are the ones that people tend to study quite a bit. We try to add standard gene and protein annotation to those records so that there's all the protein content is represented there. And they also have names that people can refer to. So they're all using the same language. So I wanted to discuss an example of how RefSeqs play a role in public health responses. So very early in 2020, GenBank team notified the virus team that they had received one of the sequences for the respiratory illness that was circulating in China. I was very interested in this. So we worked together to try to understand what was this. We knew that this was going to have to be a RefSeq. People were going to be interested in it. So within 24 hours of release of the public sequence through GenBank, we also had a RefSeq sequence that was available for everyone. It had updated annotation. So I looked at related viruses and tried to find the annotation that looked most believable, had the most evidence. I also reached out to experts in the field and just really quickly try to get as much information on appropriate protein names, start positions, anything else that was important to annotate on the RefSeq. This also was a way to demonstrate the SARS-CoV-2 taxonomy. So as people start submitting sequences, they need to know what tax group they should submit the sequences as. So this gives people a very simple way to say, oh, it goes right there. And then they can submit it a little bit easier. The RefSeq was used to create Vader models, and Vader is a tool that we've developed to do annotation and validation of sequences. We did this in collaboration also with the GenBank submission team, and this is to help facilitate submission of sequences. So Vader basically, so it takes the annotation from a reference, in this case from the RefSeq, and applies it to sequences. As it does that, it's looking for things like stop codons or missing ribosomal slippage sites, things that we think are important on the viruses. That's the validation that happens. So normally in the past, submissions to GenBank were manually reviewed. It could take months from the time that you sent a sequence to the time that it was publicly available. Having an automated tool remove the requirement for manual review of the sequences, which means within an hour, your sequence could be out to the public. And I think that was really critical. I hope that was really critical to the response, the public health response for SARS-CoV-2. Another thing that was really important was that it provides widely available sequence coordinates. So I think everybody in this room, if we say SD614G, we know exactly what we're talking about. It doesn't matter if there's an indel early in the sequence. It doesn't matter what country we're from. We all know what SD614G means. So I think that's another really critical role, that having an early RefSeq available that everybody can use as a reference is really important. Okay, back to the NCBI virus results table. So on the left side of the image, there's a list of filters. So you can choose as many of the filters as you would like to apply for a particular search. You can search by things like the accession, geographic region, submitting authors, the host names, and the host is mapped to taxonomy. So you could search for dog or you can search for mammal. There's lots of filters here. I hope if you're interested in things like this that you will go and play around with it. One of the things that we also added during the pandemic was pango lineages. We were able to add this because there's an automated tool available. Otherwise, we would not have been able to provide it. And I think it was really critical that we were able to provide it. So every evening, we check for updates to the tools, and then we run all SARS-CoV-2 sequences through the tools to make sure that all of the sequences have updated lineages as the definitions are changing over time. So that supports filtering by lineage. It also supports when you do a download, that you can download the lineage itself, and you can download the versions of the tools that were used most recently to assign the lineage to that sequence. That gets long. So there's a example on the bottom of how the versions are provided in the download. So it has like each step, which I don't even know. I'm not sure if we're using all of these. We're definitely using Usher in here. But it provides them so that you know what the most recent version of the tool was. Okay, NCBI taxonomy is also a very important part of this conversation. So we've heard how the virus species is defined by the ICTV. That gives us a very formalized way to talk about virus groups. Excellent. There's also virus names, common names, things like Nipah virus or SARS-CoV-2. We know what we're talking about, but there's no like firm definition of what these mean. When there's no official taxonomy available yet, NCBI is still receiving sequence data, and we need to put a name on it somehow, right? So we are placing sequence records into what are called unclassified bins. Conrad is here. If you have more questions afterwards, Conrad would be the person to speak to. NCBI taxonomy also includes synonyms, virus names. I think it includes common misspellings. Okay. Common names of the viruses, all of this to support making data search easier. And then all of that official taxonomy plus the accoutrement of taxonomy, they get included in NCBI virus. So you can use any of those terms when you're searching for sequences. And it makes it easier when you're maybe still trying to learn the new binomial names of species as they're emerging. It also includes nodes below the species level. So this is obviously why we're here. The subspecies classification is not organized by anyone in particular just yet. So the names are available, but I highly encourage people to review what they expect to find in there. So I love the NCBI taxonomy browser. The easiest way to find this is really to like just Google or do a browser search for NCBI taxonomy browser. It takes you to a hierarchical tree, so you can do a search for whatever you're looking for. There's like breadcrumbs from the base of the virus tree down to narrower and narrower taxonomy. If you hover over any of the terms, you can see what rank you are at. So you can see if it's a genus or a species, or it'll say no rank if you're below the species level. It also shows the unclassified bins. So this is a little bit hard to read here, but underneath of orthoparamyxovirinae, you have Nipah virus. And underneath of Nipah virus, there is unclassified Nipah virus. These are sequences that we can tell they belong in this genus. We don't know what the species is. It hasn't been officially defined by the ICTV yet. So we get it as close to the correct taxonomy as possible, but it's still in this unclassified bin. And then when the official taxonomy is released, it'll be moved to the appropriate place of the hierarchical tree. There's also a new NCBI taxonomy tree that is coming soon. I encourage people to try it out. This is the same thing, just Google data sets, NCBI data sets, and then look for the taxonomy. I just wanted to let everyone know, because I think that in virus world, we tend to deal with nucleotide records. These show the genomes, which is different from nucleotide records, but you can get there if you click on the numbers. All of our pages have yellow feedback buttons at the bottom, and we really do, we read all of the feedback that we get. I mentioned that you can search with any level of the taxonomy in NCBI virus through the virus filter. So there's some examples here where you can, you really can search with a lot. And then the results are organized by the taxonomy names that have the most nucleotide records associated with them at the top, which I think is a good way of finding the place where you actually want to be. We also have a resource. It's linked to the same resource. So you can see a dashboard to actually visualize the data. So once you apply your filters, right, you've narrowed down your data set, but maybe you want to see if there's bias based off of geographic location or when they were collected, because we know like funding and efforts are variable across time. So the dashboard's a really nice way to visualize it, and it also functions as filtering, so you can change things around and see if you can improve the data set you're working with. I mentioned the flexible download formats, so you can get FASTA. With FASTA, you can customize the def line. So you can include like whatever fields you want in whatever order you want in the def line. As somebody who used to make a lot of trees, that was very important to me. Also in the results table, if you download the metadata table, you can include which fields you want in that table. And we have randomized subsets. So this is a way of getting a smaller download that is hopefully still representative of the larger diversity of whatever filtered set you've already got. We have a new tool called NCBI dataset. So you can use this through command line or API to get the data that's available in NCBI virus. So the curated data with the taxonomy, but it's programmatic. So you can put it into a pipeline. If you want large data downloads, this is the way to go. And I put the URL up there, but really the easiest way to find it is browser search. There's a theme here. Okay, I think this is close to my last slide. I'm asking to help us help you. When it comes to subspecies classification, we know that the lineages can change over the time, but the data that is provided during submission is archival. That means the only people who can change it are the submitters of the data. So over time, as the lineages are changing, if it went there, it's going to stay static. So a good place to put classification information might be NCBI virus, but we need an automated way to get the lineages. And we can help to distribute the lineage references also, as long as they are like not consensus, but actual records. In order to do this, we need to have community accepted classification schemes. So wide levels of acceptance to show that there is actual desire and interest in having the data available. And we also need the automated classification tools that have some plan for long-term support. Because if we put something in place, and it takes quite a bit of effort for us to put something in place, and then 6 months down the road, there's no one to support it, then everybody's going to be upset, right? We don't want anybody upset. With that, I would very much like to thank the entire NCBI virus team, including 2 former members who are here, Rodney and Ryan. And you've heard Rodney's name throughout like so many of the talks yesterday. We really like to play a role in helping to share data and make it accessible to everybody. Also, our NCBI and NLM colleagues and everyone who's contributed to the shared public data. And I think that is it. My email's on here if you have any questions or suggestions. Thank you very much.
Our next speaker is Philippe Mercier. He's going to talk about virus subspecies in ViralZone and UniProtKB.
Okay, thank you for inviting me. So I'm Philippe Mercier from Swiss Institute of Bioinformatics. So today I will talk about what we have in subspecies in ViralZone and UniProtKB. So ViralZone is mostly a knowledge repository in which we have sheets for every kind of viruses, from phages to human pathogenic viruses. And this kind of put piece of puzzles together, those sequence and knowledge we find in books. And we are linking, for example, to NCBI virus on many resources. We are updating the link to BV-BRC. And of course, so I will talk about the variant in ViralZone, what is in UniProt, and some thoughts about variant annotation or metrics. So we have a ViralZone resource for different pathogenic viruses like HIV, SARS-CoV-2, HPV, herpes, pox viruses. So we make a focus in these viruses much more than in others. For example, proteomes or interactome, I mean the interactome, the curated interactome, not the large-scale interactomes. Life cycle, with their antiviral drugs, and we made leaks to Nextgen and many other resources for phylogenetics. So this is kind of a toolkit for when there is an epidemic or something, many labs switch to, or students want to know more about the virus, and they need to have a basic molecular biology about it. So this is a kind of start for them. So this is a monkeypox resource, pretty much the same. I will not go through it. So, of course, there are many pictures in ViralZone. They are now in SVG, high resolution format. It's free to use. It's a CC BY, just have to name. There is no copyright, hopefully, on the picture right now. Everybody can use it. It's free. So we have, we put variants on genome pictures for everyone. So for special virus, like having polyproteins, so the topology and cleavage are indicated in a way that is more convenient than in a file. So some curated interactome shows a main interaction that have a functional effect, proven effect. And of course, a different life cycle for each viruses, okay? And in this life cycle, we put when there is some the antiviral drugs and which point they are acting on which one. So for SARS-CoV-2, it was a bit a mess because a lot of going on and off. Does it work or not? So on. So when we release, for example, the resource for coronavirus, there were a lot of people that use it. So you see compared in orange the usual users, and in 2020 in blue you have much more. So people were really kind of happy to access this kind of data. Even I had some people from UK politics of parliament asking some questions. I never know where Zone would be read by such people. I think it was for scientists, but so it reached a lot of people, which is nice. Of course, in early 2021, the game changed a bit, and there was a variance coming up and taking a lot of place. So we started to create a variance page, which are not links to Pango, covariant, outbreak info, and BV-BRC resources. So it lists the main circulating variants, where the mutations and in kind of graphics. And we have also a list in which you can find every data. So you got the links to Pango, covariant, and so on. Especially what user wanted to get some consensus sequence, some genome or spike protein FASTA to make many analyses. For example, people making a prediction of 3D models and need a sequence. So that's where we have been fishing very early for reference sequence for this. It was not very easy at first. So, so for example, for choosing the right mutation, now we have this resource, which is pretty nice. You can see, you can ponder how many mutations there are within Omicron and so on. So it's outbreak info. In the very early days, I used CoveThread 3D, in which it was very convenient. You can put a mutation or several mutations, and it gives you everything in GenBank that correspond to that for SARS-CoV-2. So the first variant alpha and beta, it allows me to find actually sequence, and I took the sequence, make a tree, and choose which would be the first one. So later on, we have discussion between BV-BRC and NCBI, NIAID people to select the sequence and to present the same references. So, and so in UniProt also, there was the idea of putting variants because users wanted to get access to this data. But UniProt is a bit like the Titanic when it comes to agility. And I mean, Eneida, it was 24 hours to get the sequence out, but when the sequence was out, I actually picked up, and within 2 days, I annotated for UniProt of the SARS-CoV-2. But then they told me, it's not going to be released before April or May. So we were in January, okay? So it's not that quick. The problem is that the structure of UniProt is that you release everything at once. You cannot release one entry at all. It's not possible. They don't want to hear about that. So they created a special resource for SARS-CoV-2, which is COVID-19 uniprot.org, in which we put everything that we are able to update on the fly, like in 24 hours, just on this resource for users to get access to early annotation. And for variants, so UniProt has a big problem of having to run a lot of prediction tools. It takes a long time, so they tend to reduce the number of sequences they have. So, for example, there is a debate now. We will just take one sequence per species, per ICTV species, presumably in UniProt. The others will be put in UniPark or other stuff. And we are currently trying to use various exemplar for ICTV, but it's not yet decided. So I could not create different entries for the variants. So we have to put in the main entries in kind of feature variants. And actually, many users were happy with that. So I'm surprised because it's not very easy to work with, but many people using UniProt are used to that kind of annotation. We find that some nomenclature are not very consistent. For example, if you look at break info for BA1, they say that N2 to 11I, and the deletion is 2 to 12, but it's not in BV-BRC or in covariance. So we're asking what happens here. So I mailed to Emma because we're working together with Richard for Swiss surveillance. And she told me that BV-BRC and covariance seems to be best choice. So at the end, I took a Swiss decision. So stay neutral. I said, I put NL turns to a high, but maybe we could profit from some kind of standards for this kind of annotations to avoid these discrepancies. Okay. So now about annotation. So we have made a lot of progress to produce the sequence to exploit, to classify that over the past 20 years. It's incredible how fast or good it is. But for predicting the phenotypes, we did not make so much progress, actually. So ViralZone now produce a lot of sequence, but there is not much power or even money to analyze the sequence and to make sense of it. We have a lot of full genome sequence, and we are not really able to take all the information which is in it. There have been, in SARS-CoV-2, the main focus of publication has been on the ACE2 binding. So it's like everything in the virus is turned into, you bind more to ACE2, so you get a better virus, which is much more complicated than that. For example, underreported was the deletions with the stop codon in open reading frame 8. So you're losing a gene which is supposed to be virulent, like monkeypox is losing some genes or pox viruses. And pretty much nobody talk about it, just focus on the ACE2 binding when there are many other ways to analyze. Also, in virology, glycosylation plays a much important role in antigenicity, and many viruses play with that. In influenza is famous, especially with H3N2. So you have here different influenza hemagglutinin, and you see how glycosylation in yellow is changing over the years and over the different subtypes. Also, HIV, for example, we call that a glycan shield. There's a lot of glycan that move a lot and also participate to the escape of the virus to antigenicity. So I thought I was smart to look at glycosylation in SARS-CoV-2 for every variant, but actually it did not change at all. I mean, it's like the virus doesn't care, doesn't need to change it. So except for one minor variant, there was one glycan that was supposed to be escape anyway. So I use a glycan prediction tool for that. So and you see it wasn't important for SARS-CoV-2, but I think it's still, the idea should be there to look at for any other viruses. because that's really a way that viruses are modifying their antigenicity. Okay, also, and we have seen with this plot that there was a lot of disordered regions in the spike protein, for example, on nucleoprotein, on many variants before Omicron. Variations turn out to be present in these disordered regions, so there may be something about antigenicity there as well. Actually, many people thought that the spike was fully structured because in PDB, you don't see disordered regions, so people were believing there's no disorder at all. Hopefully. And you know, AlphaFold is showing up the loops that should not be ordered. And you see that AlphaFold database does not make that, so we need to improve there as well. And you see, and of course, something that users may need is metrics for genomic heterogeneity. I've heard a lot of people in the past days saying that my virus is very diverse, not so diverse, and so on. But it would be nice to put numbers on this to be able to compare. So there are some tools like PASC already been told in many speeches before, and Vista is doing the same, in which you can actually look at below species diversity and you have a graphical view. You can see how much is the diversity there, but it's not a number. We have conducted a study in early 2020 for human pathogenic viruses. You see the complete genome sequence on the left. You see influenza virus, we have much more sequence than HIV, for example, but when you cluster the sequence to 98% similarity, you see HIV is as diverse as influenza A. And when you cluster at 90%, then influenza collapses, but HIV stays very diverse, actually. So there must be a way to ponder this diversity, to be able to compare them. I think it would be interesting even below species. And I was interested by NG, you have a genotype, representative index, something like that. That seems to do the job. So we'll see later. Okay, and about representation of data to do the job. I just wanted to say that PCA, principal component analysis, could be interesting for many below species analysis. You see, we made that in 2008 for to do the Z epiflu that we made. So the PCA analysis here, this is the HA. You see, it's colored by year, and you see clearly the genetic drift of human HA from the 33 where we have sequences. You see the 2009, and you see also different points that tells you that maybe there is some sort of recombination error, whatever. A recombinance in PCA will be between two kind of dots. So maybe it will be interesting. And I just highlight that PCA is used by many people where they don't have sequence actually for classification. Okay, so in the conclusion, I want to just put to do the top structure of the different users' needs and the resource we have. So for classification, people need a repository database. So we have an SSDC, GenBank, we have GZ doing really nice work that they organize the data on the metadata, and they check for the quality. Then we need classification system. So there are many classification systems. It depends, of course, of the virus. It was interesting to see that Richard pointed out for RSV could use the Pongo system because it was nice. So maybe we could have some recommendation which kind of system would be the best for the future classification. Then for users, it's need automatic classification tools. So fortunately, I heard today that there are a lot of classification tools around automatic to do the nice, because otherwise, if you just make a classification system you publish, but you don't make the tool, it will not be much used. And classification tool, as you pointed in Eneida, you can make it back to a repository database and classify everything inside. And that's really what users need, because then it's very clear. So there are Pangolin, Netscape, there are BV-BRC, but there are many tools developed in, for example, Oxford Bioinformatics. So I try to link that in ViralZone. And then all this classification system works very well now, but then users need more, for example, for pathology, when there is an epidemic, the doctor wants to know, now they can have a very early sequence of the virus, so they can really treat the patient to do the job between it. So there's a drug resistance, which is not obviously very needed for SARS-CoV-2, but for HIV, for example, it works very well. People are looking for drug resistance and adaptive to treatment based on the sequence, and to do the influenza as well. And for any future epidemics, it could be nice to be able to predict very early drug resistance if there's drug and if you know it. To do the job. Also, antigenicity is very important for hospitals because, for example, we have monoclonal antibodies for treatments, and when there is a new variant, people want to know if the monoclonal antibody will be efficient or not. So this is not able to predict right now, actually. I hope it will be in the future with 3D modeling on large language model. But so far, to do the is more on experimental data, so it's a bit longer to be able to predict that. So up to the future, we'll be able to include that into the loop as well. And then there is a tropism also, which is kind of when the virus switch between different receptors or species. So we could have a lot of this with the global warming on arboviruses that are evolving a lot. Again, 3D modeling can help to find a change in the receptor binding, to do the, of course, experimental, so on. To do the job, some kind of analysis for epidemiology, epidemic models, and a lot of things. So I may be missing a lot just to put some things in place. And at the end, you have some knowledge repository, which are PubMed, of course, UniProt, ViralZone, BV-BRC. So to do the job, I think a lot of people in the Swiss-Prot for genome annotation and ViralZone, the collaboration with the hospital in Geneva and with people in the surveillance like Richard Hemmercroft, people in NCBI, Los Angeles for reference sequence choice, and people in NIH in Florida for the pox virus data. Thank you very much.
Our next speaker is Andrew Warren from University of Virginia, BV-BRC, thinking about virus mutation spectra through visualization.
I did change my title a little bit. I took spectra out of there because I thought it might be a little confusing. I don't have any mass spectra or everything quite so fancy. So when I was talking to Elliot about this workshop, he asked that I sort of look at visualization tools that support working with evolutionary lineages. And so I want to kind of go into briefly what this talk covers and what it doesn't. I'm going to try to give a sense of different scales of classification using different visualization examples, and talk about how those scales are applied, examples of tools that leverage those, and sort of give us general food for thought using visualization that touches on many of the discussions that I think we've had throughout this workshop. Sort of call back to the dimensionality aspect that Tavis brought up with respect to visualization. You can't really do a visualization unless you have unless people can actually use the tool, right? And the data is available, and you can load the data into the tool, and they can understand how to use it. There's no point in using the tool unless there's specific questions to be answered. No one will want to use your tool if you don't follow some sort of information visualization principles relating to information, like PAC and sort of Edward Tufte's school of thought, and interactivity and availability of data. All are elements here and you can't divorce them from these visualizations, right? It seems like and if I did, it would be probably a pretty boring talk for this audience.
To do the job.
And so this applications idea harkens back to what systems of classification can be used for. I think we had some discussion about this yesterday, whether you using them for communication to sort of support the attention function after you create a new pangolin lineage so that it enters the dialogue of people's attention and say, oh, okay, this B117 or something XBB 1.9 is something that we see growing in India or wherever it might be, and then there's analysis, which isn't necessarily right. It is often I think in the case of many classification systems, it's conflated. People will do analysis on particular namespaces, but whether that namespace was meant for that or not, right? And it's hard to realize and think about that. And I just want to point that out as we go through some of these visualizations. And another idea about what you can use classification systems for is quality control. And you can argue this is a form of analysis, but in terms of sort of stability, understanding differences in calls between different systems, having a namespace provides that sort of common language to sort of characterize in a parallel system. If you think about a phylogenetic tree existing and a pangolin system existing, having the binding between those really allows you to kind of review the resolution of calls that are being made as you go on through time, and there are virtues there that I think have been well described. And I think one tenet that's important here is that the analytical abilities and visualizations that are enabled often depend on the level of sequencing associated with the organism. I think we made that point yesterday with rotavirus and others. And usually complex questions and visualizations actually require work. There's not too many tools that are just going to, you're just going to show up and say, I want to know this, and it's just going to magically give you, everything you want. And I put this quote down at the bottom because I really liked it. I read this paper last week in trying to get ready for this thing. And I think it's more broadly applicable than the difference between phylogenetic placement and machine learning methods, in that, attaching these naming systems to phylogenetic trees really provides a form of regularization that provides stability over time and allows us to think about them, which I think, again, goes to the communication facet. I can't remember if this graph shows up on the next page, but that's a genome graph, which is essentially a glorified multiple sequence alignment. And it's a multiple sequence alignment that puts on errors, I guess. And it has explicit modeling of breakpoints and of haplotype structures in that. And so it's not laid out in a nice rectilinear way because it's hard to do that with a graph. But I want to maybe we'll get into it in a little bit, but I'll offer that as in terms of these challenges faced by HIV and other things where recombination is an issue, starting with, instead of a multiple sequence alignment, which enforces that rectilinear arrangement, having an explicit model that in the data that deals with that has a lot of advantages. There's been a lot of work in recent years and months related to higher order organisms. You might be familiar with like the Human Pangenome Initiative work, and work also in bacteria that uses genome graphs. So that's my proselytizing for genome graphs, right? For those of you that haven't encountered that already. All right, high level markers. So this is a binary classification system right here, SGTF, right? The spike gene target failure. And in conversations previously, people are like, well, it's just a piece artifact of a PCR primer. Well, it's one that keeps showing up on a regular basis, sort of interestingly pointed out in this MMWR paper. And then on the right there, I just I did a query and took from Cov Spectrum, and which I'll talk about a little bit more in a minute, for S gene dropout, and it continues on past the end of that red graph hopefully you can see over there that actually it's now reached sort of its zenith, and in terms of the virus exploring different sort of macro level solution spaces, you might anticipate a toggle back down again, sort of a ongoing mystery that I think is interesting. Okay, so pangolin lineage, where this is an example from UKHSA, where they're paying attention to the distribution of pangolin lineages at a particular point in time. So time is just as a footnote to the image itself, but you can actually see that the width of the Sankey diagram here and the percentages associated with these different names are actually, I think, quite extraordinarily distributed among this namespace for this time and place, right? So normally when people do these analyses in mini plots, in the stack plots that you often see, you don't get to see, they dedicate the space to time, and you don't get to see the actual breakdown. And so I think this highlights well there's this global attention function for creating namespaces, especially in subspecies classification when we're starting to do genomic surveillance more actively, and to understand what's going on with disease spread. Having pangolin has been tremendous, so you can actually, again, have the conversation, have something to point at and say, okay, it's stable across everybody's trees and so on. But it's really pretty interesting that you can ask the question, do we need all of these in any given, even in this plot, right? Like, are they all meaningful? And I don't have an answer. I just think the question is important, right? Okay, and so on the left, there's a plot from it's one of the Genomium Theo Sanderson enabled tools, and I thought I had a link on there. But this shows the nucleotide distance between different pangolin lineages, right? And so that again just emphasizes that the granularity in the distance between the lineages is non-uniform, and so when people are starting to analyze them and say, okay, this thing has immune escape, or here are all these co-circulating variants, they may or may not, always have explicit meaning relative to the spike protein, to antigenic escape, and be I don't want to say they're not worthy of being in the conversation, because you often don't know a priori which thing is going to become important, right? And so, but it's just something to think about in terms of this namespace. And then on the right, it has to do there with this upset plot has to do with again a moment in time, a snapshot of the differences between pangolin calls between GISAID and next strain. It was during one particular week. And pangolin lineages are being updated, right? So often what you'll see, and I apologize, it's a little too small to read, but if you all get up out of your seats and come to the screen, you'll see that often XBB 1.5 has a relationship to XBB 1.5.33. Like, those are the differences between, and it just has to do with pangolin, let's say pangolin deployment, and sometimes there's sort of jarring cross-lineage reassignments. And I just wanted to make the point that this stuff is knowable and has been a challenge for different people at different times, associated with an actively used naming system as part of the as part of genomic surveillance. And so if you do away with or you want to introduce the element of time here, this is just a spatiotemporal analysis frame. And I'm going to pick on the COVID data tracker for CDC here, where there's a challenge, to decide, again, how many of these different lineage names need to be in there, how fine-grained are you going to pay attention in terms of this thing. And there's some work we have on that in terms of parameterizing that, making it for a given time frame, allowing a system to decide, let's sort of do a principal component analysis to say, and we're in the process of validating this, but how important are these variant labels, or how likely are they to be represent like distinct portions of the antigenic landscape and claim their fair share of infections going forward? I recently, when Betty recently started, or a few months ago, started giving talks, regular talks to Spears, so I discovered the LANL site. Again, this is an example of these stack plots that really allow you to track what's going on globally and more locally. And then this circle plot on the right is just, again, to emphasize the fact that there are all of these contributors. So if you see JN1 multiple times, trees contributing information. I'm not used to that problem. Okay, so this is again another level of taxonomic. Of sort of hierarchical organization where people started paying attention to convergent evolution. They started paying attention to how much immunoscape the number of mutations likely implied during the sort of BA5 era, and they started naming these things, right? Like level 1, level 2, level 3, level 4, level 5. And that was a classification system that talked about immunoscape, right? And some people found it more useful. And I just want to point out again that this existed and people were doing this, not because they were bored, right? It was because it simplified ways to talk about that. And it may only be manifest in viruses that are well surveilled enough to actually identify that this is going on. But still, I think it's an interesting level of organization. Okay, and sort of the smallest level of organization I could think of is paying attention to an infection cascade in an individual location. So if you one of Angie's lab's group's tools is Cluster Tracker, which pays attention. It takes the preponderance of all surveillance, and then it identifies, like say in the United States or even globally, it identifies nearest neighbors for regions of interest, say your state or county or whatever, and then from it calculates an index and it's able to determine which things are importations. And through this repeated calculation, you can put all of these sequences together and have a sort of sketch of a contact tracing, sort of mesoscale contact tracing, which is visualized in the upper right. And so that is a really fine-grained level of organization. They all have cluster IDs, right? So again, these are things that are just sort of different, really small fragments of the phylogenetic tree, rather than the really large segments that we saw at the beginning with SGTF, right? So, okay, this is the slide that I use. I'm going to use to razz everybody a little bit. So this is I can't have a visualization talk without having bad visualizations in terms of this overly complicated Venn diagram. And then also there were people were reassigning, these lineage names to Greek mythology at one point. Again, I think for communication purposes, right? And it was effective in that, and I don't think anybody got any particular credit, like I named this thing Cerberus. But it did gain additional interest, which was, potentially the point. And again, here is an example of this number of changes for the convergent evolution there. Okay, so now I'm going to pepper in. So now I've covered some sort of spectrum of hierarchical organizational schemes. I'm going to talk about a few different useful tools. These are in no way it's an impossible task to characterize the universe of tools available to us, and all visualizations available to us. So this is just a smattering, so that I can fulfill my duty. So useful tools, Archaeopteryx in the MSA viewer in BV-BRC. It's also available as a download. It's one of the benefits of BV-BRC. One of the strengths is the different types of metadata that you can bring in and pretty easily associate with different visualizations, and interact with these diagrams, make different groups of things and take them away and do whatever you're going to do. It talked a lot about NextStrain, NextClade. Cornelius highlighted the recent addition of the forecasts to that environment. I think it's a wonderful tool. One, I guess, added detail is that Angie told me about earlier is that they have the ability to view mutation annotated trees, right? Which is a relatively new thing in the pantheon of phylogenetic phylogenetics and support for things, so you can actually see the differentiating mutations associated with the tree. Cov Spectrum, even though this isn't overly graphical, the data model for Cov Spectrum is fantastic. I think there's a CDC project right now to expand the data model to other viruses. It has a very, very powerful data model for doing querying associated with the different things. Trenome, Taxonomy. This is again in sort of the Theo Sanderson part of the world. It scales very well. It's a web-based tool, so it's easy to go to. Has dynamic loading, so it supports tremendously huge trees. And this is this Trenome thing is a coordinated view, so you can look at the annotations, sort of get a sense of the multiple sequence alignment all in one, and go in and interact with things. This is a tool that's very obscure. It's from a Helsinki University master's thesis, but it was quite a number of years ago. It was one of the first to very support sort of dynamic scaling for very large trees, and you can zoom way out. And it has a coordinated view, it has good interactivity for selecting columns and things. Okay, so I'm going to talk about potential gaps, mostly to prime you guys to think about potential gaps in terms of tools available to what you would like to see. I think there's a lot of stakeholders here that are sensitive to feedback about additional capabilities. So epistatic interactions, I've seen enough, again, of Betty's talks to think about, or at least she's, unless my intuition is wrong, to prime me to think about haplotypes and whether you want to call them, quasi-species haplotypes, epistatic interactions, linkage disequilibrium, all these sort of phenomena that have to do with mutations that impact other mutations. I don't know the level of surveillance necessary to actually actively investigate that. Certainly I think SARS-CoV-2 has achieved that, probably influenza. I don't know below that whether there's sufficient data to achieve good p-values associated with that. But there's examples of the sort of a famous example in Omicron was a compensatory mutation that allowed additional degrees of freedom in spike. And then let's see, I'm running low on time, so I'm going to skip some of this. This is a call out to the NCBI Codeathon that we did not too long ago between NCBI and NIAID. And just to highlight the result here, because I happen to be sitting next to Ryan yesterday, and he's the guy that's supplying the data. He was very kindly gave me new data associated with looking at coordinated mutations in SARS-CoV-2. So that's a network of all the coordinated mutations with significant Pearson correlation coefficients. And I looked at SGTF, and he pulled down, he did the query for me. So he looked at SGTF. And here is a list of mutations that are significantly, have really high Pearson correlation coefficients associated with S gene dropout. So there's potentially an epistatic network there that's manifest, is responsible for this toggling, right? Back and forth. And I think the top one there is present in 8% of all samples. Sorry. And this is another potential gap. I think there could be more tools that take our reference-guided mutation language and decorate structures and understand, if you have some set of mutations you're interested in, some tuple of mutations, you want to understand how they relate to each other in the structure. I think that in this tool was pointed out to me by Brian Hilsner. So BV-BRC team, some of this work is funded by those sponsors there. I want to thank Elliot for inviting me, and everyone else listed here, and all of you. Thank you.
Now we will do 15-minute question. Christian, if you can watch for the online question and read out if there are any. And any questions from audience?
Okay, so
Indresh.
Hi, Rachel Cahoon, University of Edinburgh. I think I'm aiming this more at Philippe and Andrew. Within the pandemic, we saw lots of tools that were command line, lots of tools that were done in the web interface, which tended to reach different audiences. Do you think going forwards we will continue to need both for the same tools or not?
That's a tough question because very often these different tools offer different perspective on the same data. So, to me, it's the users that will decide. Eventually, maybe users will go more to one tool than the others, and then you know that the one which is more designed for users. I think it's difficult to decide by yourself which one is the best. Well, that's what I would say. What do you think?
Yeah, I think one sort of example that might influence things is I recently had to get some very obscure set of conditions for sequences out of SRA or I think SRA, and I asked ChatGPT to generate a query for the NCBI API, and it generated it. And I said, I want it in tab delimited format, et cetera, which primes nicely the use of Excel and so on. So there you have much more empowerment for low-level access to the data, which gives flexibility. But we've always tried to hold that dichotomy. Like in some cases, you have an API, like lots of people like command line tools that actually allow you to get in there and access the data. And it can be very challenging with so many data to do web applications well that scale, in a way, and both allow them to see quote unquote all the data and allow, narrowing down. And so I think it won't go away. I think those will both persist. I don't know if maybe the balance will shift a little bit given that additional empowerment. But so
Can I add on? So we had actually quite a lot of discussions at NCBI about web-based tools versus like command line tools and how to access data. Because especially when people were repeating analyses over and over and over and over time, having automated tools is pretty critical to support those kinds of analyses. But at the same time, they are not the best for somebody who's not so familiar with something like data science or data analysis and how to explore the data, validate it, identify biases. When you have something as diverse, not as the sequences themselves, but as the submitters who are providing the sequences, being able to validate and look through them and see what you actually have before you do an analysis is important to getting good results.
Richard?
So just to follow up on that, so NCBI virus is really a great resource. We use it all the time. Both the CLI and the web interface. And in fact, we are sort of hoping to, I'm not having a question, it's just a comment. We are hoping to have more next strain analysis sort of updated every week. And without, NCBI virus, we wouldn't be able to do that, right? So here we use the dataset command to pull in the data and run the analysis of that.
We're happy to hear that. Thank you.
So Eddie?
This is for Eneida. I just am a little confused. I don't understand how you handle the ownership of, say, a Pango lineage name maybe by a submitter, and then the Pango lineage advances and there's a new lineage, and so this one should be reassigned. How do you guys handle that situation?
So there's not a particular place for something like Pango lineage in the INSDC format for submitting. So they could do something like isolate or strain, and they could put the name in there. But the way that we do it at NCBI virus, because we're not archival, we can take the data that's there and we can layer on top improved data. So on our side, we add that improved data, and that's what we provide through our interface. People can always go back to the GenBank records and get the original data if that's what they're looking for. Yeah.
Christian?
So the online questions. So there's actually, so far there are 5 of them. So just going through them one by one. So the first one is for Eneida. So it says, it's from Sushata Rashid, and it is, in metadata fields, can NCBI insist on passage history for the virus information, as that is quite irrelevant? And also make it more visible, as in GISAID. As an example, P0 for sequence from the clinical sample, P1 for first passage, etc.
So I'll answer this in 2 ways. One, it's sort of over my pay grade. I will pass the comments along. Two, something that's really interesting with passage history, because we've looked into that to see if we could normalize the information that is provided. A lot of people tend to use abbreviations for passage history, and trying to interpret what is meant by those abbreviations is difficult. So I would ask that maybe there's information so we can provide it in a more unified way.
So I'll add to that question. IDPCC, they have a data standard how to name the passages. Probably we can piggyback on that standard if NCBI adopts it. Also, I think there's a lot of that data comes in the comment. So people have free form of specifying. So providing a structured way and a specific field will help. Right, Cornelius?
Yeah, I just wanted to say it would be great if that comment also ended up in the NCBI datasets export. So we can put it in and show it on next entries.
All right, Rodney.
All right, you guys open up the door to this garbage can. So the bottom line is the way INSDC records are sort of formatted is actually not a great way to submit deep metadata like passage history. And so the INSDC came up with a different database to store this information called biosample. And so this is when I put my SRA hat on. So all of you who are submitting data to SRA are already adding this kind of information as part of the biosample package, or have at least the ability to do that. And it would be my dream is one that where important communities, and I think all the scientific communities that submit are important in a way, but certainly communities that really rely on this deep metadata, if we can start adapting standards where everybody submits biosamples, where you can have 200 fields of metadata if you wish, and then Ryan here can figure out how to actually sort it, normalize it, make it useful to people. But I think that's really the answer. And I think this is sort of a theme of mine. You know, there's some creations that go back 20 years, 30 years that we're using today to do modern stuff. And at some point we have to say, these just aren't what we need. And we need to start using the things that we need. And I think biosample falls in that list because it allows you to have fairly standardized packages that everyone in the community can use, as well as add all this additional metadata.
I completely agree, and I've been begging for it. All right, online question.
Yes. So the next question is also for Eneida. It's from Priya Onoda, and she asks, heavy user of NCBI platform. Thank you. My question is, what is the current requirement to be called a reference genome? Does it get sequenced multiple times to account for sequencing error, or something like a pan genome, which would be ideal?
So in most cases, RefSeqs are selected from the ICTV exemplars, where the study groups are looking at selecting the highest quality examples. And we do some validation on the ICTV exemplars to make sure that they are records that we can promote to RefSeq. But in general, we take their recommendations. For things that we create sort of from scratch as a RefSeq, we tend to do things like read the associated publications, look at what the sequencing methods were, try to see if there are other similar sequences that we can compare it to, look at how much metadata is associated with the sequences, just any indicators that we can find of potential problems versus potential quality of the sequence when we're choosing the RefSeq.
Let's take one more online question.
Okay, one more. So that's from Ruiming Gall from the Public Health Agency of Canada. And that person asked, if there are certain duplicated genome in both NCBI and GISAID, for instance, are there any resources available to link both accession numbers? I'm not sure for who it's for. Maybe for.
I've heard rumor that there are, but I don't know what they are.
Good answer.
All right, Angie.
At the risk of getting in trouble, you can email Angie at soe.ucsc.edu.
Good luck, Angie.
Richard.
So, Philippe, I know that in order to generate these pages for the different viruses takes a lot of manual curation and reading of the literature and stuff like that. Are you guys exploring using Gen AI to like give initial drafts of the content for these pages, or?
Well, we use a lot of resource to get the content of the resource actually. But mainly from PubMed, we go back to the source of every knowledge and look at it. Sometimes we don't find it, so it means that people are talking about something that's not really sure. But we are really checking the quality of everything, pretty much from PubMed, also from books, and also asking experts. So, for example, for the pox virus, it's quite complicated, and that outside of my usual knowledge of negative strand background. So we have contacted experts and we submit what we end up with to them, and they correct it. So to get the best quality at the end.
All right, let's take one more question. Okay, Liliana, go ahead.
I just wanted to make a comment for the panel and for everybody here of the great importance of all of the visualization and analysis tools that you're providing, both through APIs and web interfaces. I know that many countries of low resources have made a lot of use of all of these tools. And so the access that we're providing globally is really, really very important and of utmost urgency in the middle of pandemics for those countries who don't necessarily have the same tools. So continue doing them and continue exploring new ways to do this. Really encourage that.
Sure.
I just wanted to add one more thing. I hope that I made the case for the importance of RefSeqs to the community today. It's a lot of work maintaining those also. If you have a problem, if you see something wrong with your favorite virus's RefSeq, please reach out to us because we are very happy to make the improvements to make them better for the communities.
All right, we have 1 minute. All right, Richard, go ahead.
Yeah, Richard, yeah. I have maybe one final question, and that pertains to making submission easier, especially when I hear sort of the requirement of adding SRA with 200 fields and so forth, right? This might make the, if successful, the data in the database better and richer, but might sort of increase the barrier to getting it in there in the first place. So, are there any sort of maybe sort of generalized, sort of generic initiatives to have pathogen-specific submission?
So again, I'm not part of the GenBank submission team, but I do try to work with them to try to develop tools that make submission easier. One of the things that I mentioned was a tool called Vader. It's a viral annotation definer. Because those have validated annotation and they have the types of validation that GenBank applies to sequences to try to keep the data in GenBank high quality, those let things go through much more easily. So they're actually working on quite a few improvements to support the submission process and make it easier for people, make it faster, make it simpler, make it easier if you're a new user of it. But I'm definitely hoping to continue the collaboration that we have with them to increase the number of sequences that can just slide right through, because it has a Vader annotation and validation step in it.
Yeah, and quickly add with submissions for the biosample will have the same requirements as GenBank. It's not you have to add 200 fields. You'll still be required to add the same, I think, 3 or 4 fields now required. It just gives the option of people adding a lot more information that they want. And a lot of communities have come together and really kind of demanded that, particularly around COVID. So, and I think Eneida deserves a lot of credit here because she's pushed forward several annotation pipelines, which are actually submissions pipelines, which make it very, very simple to submit data and very rapid to submit data. And I think that model is really an amazing one and hope to see that expanded to many other viruses.